Our presentation is on climate change and infectious disease.
Introduction. On this slide, we're going to be discussing the life cycles of 3 microbes: bacteria, viruses, and parasites. The first microbe is bacteria. The life cycle of a bacteria can be broken down into 4 stages: lag, log exponential, stationary, and death phase. During the lag phase, bacteria are adapting to the environment. The log phase is when the bacterial population grows in large amounts. They start consuming nutrients and producing waste. When the nutrients run out, the bacteria will enter the stationary phase. This is when the population stays the same. It doesn't increase or decrease. The bacteria stop growing but remain metabolically active. And the last phase is the death phase, which is pretty self-explanatory. In a virus life cycle, there's only 3 parts: entry, genome replication, and exit. Entry includes attachment, penetration, and uncoding. It then enters genome replication, where the virus relies on host translation machinery and ribosomes for their protein synthesis. This step varies from virus to virus. Exit includes capsid assembly, release, and maturation. Capsid assembly is where the capsid is assembled and the genome is packaged within. Release is where the virus particles are released into the host cell, and maturation is the determination of the infectivity of the virus. The last microbe is parasites. The life cycle of a parasite can also be broken down into 3 parts: growth, reproduction, and transmission. The growth and reproduction part of the life cycle are self-explanatory. The transmission phase is when the parasite gives the host a disease before leaving the host body, but these 3 phases can be divided into 2 categories of direct and indirect. Direct parasites stay inside of one organism and eventually leave to live in the environment around it.
Predicting the consequences of climate change on infectious agents. Climate change can benefit infectious agents in many ways. It can increase parasite growth and replication. It can increase rates of pathogen transmission due to biodiversity loss. It can increase food or waterborne illnesses due to drastic weather changes, such as increased rainfall and flooding, and it can also interfere with host immunity due to warmer temperatures and climate variability. Ecophysiology of host pathogen interactions. Host pathogen interactions describe the complex relationships between the host and the invading pathogen and how pathogens sustain themselves within the host organisms. As climate change causes temperatures to increase, both the host and the pathogens can react differently. Understanding these relationships is crucial for predicting changes in disease dynamics as temperatures increase. Studies have shown that the increased levels of parasitic growth and replication due to warmer temperatures can outweigh the increased immune function of the host. For example, warmer temperatures increase immune functions in gorgonian corals by increasing cellular and humoral defenses. But since coral pathogens also replicate faster due to warmer temperatures, there has been an increase in disease outbreaks in the Caribbean coinciding with warmer sea temperatures.
Ecophysiology of host pathogen interactions continued. The metabolic theory of ecology, or the MTE for short, helps to reduce the need for further research on species by using what is already known about the relationship of an organism's metabolism, environmental temperature, and its body size. The MTE is an approach that can be used when there is limited knowledge on a certain species. One study utilized the MTE and the traditional host parasite transmission models to analyze how changes in temperature impact the reproduction of strongylid nematodes.
Due to climate change, interactions between host and pathogens may change with new species pairings as well as a loss of known pairings. Species known to interact with certain pathogens may not interact anymore, or vice versa. Some species may interact with pathogens they didn't before. An example of this is in the Caribbean, which scientists call a disease hotspot. Coral reefs have showed a decline in the Caribbean due to new pathogen interactions as a result of the rapid warming. When a keystone species is impacted by changes or diseases induced by climate change, multiple trophic levels can be affected as well, disrupting the ecology equilibrium. An example of this is shown in seagrass declines that have been infected with the parasite as a result of warming from climate change. Once the seagrass declined, nursery habitats were lost for fish and migratory waterfowl, as well as the extinction of eelgrass limpets.
There are two different types of microbes. One is commensal, which prevent colonization of pathogens by acting on the host's immune system to induce protective responses. And there's mutualistic microbes, which are microbes where both the host and pathogen will benefit. An example of a mutualistic microbe is probably the bacteria in your stomach that help with digestion. The increase in temperature will lead to a decrease in pathogen interaction and could also lead to the extinction of host communities. Effects of biodiversity loss. When biodiversity is lost, basically there is less competition and certain parasites will have it easier than others. Phenology is the study of patterns and cycles made by living things. The summary of this slide basically says that climate change is changing behavior, movement, and phenology of hosts and parasites. An example of this is the asynchrony in the seasonal activity of larval and nymphal ticks. This will cause the Lyme bacterium to be delayed.
The consequences for conservation and human health are huge because of climate change and wildlife diseases. Climate change is causing an increase in infectious diseases which affects humans who rely on the wildlife for food. Warmer temperatures and heavy rainfall can cause cholera and other diarrheal diseases. Mosquito-borne diseases such as malaria and dengue fever have also been seen to increase because of climate change. This has huge societal implications. The crops and animals humans depend on are dying, and diseases like cholera affect millions annually and have a high fatality rate. Because of this, humans are implementing forecasting and modeling. Forecasting leads to improved timing to protect humans, crops, and nature systems from harmful things. An example of where forecasting was helpful was in Botswana. It helped protect the humans from malaria. Modeling helps to predict loss events and improve insurance returns against the loss. Forecasting and modeling are important because it will ultimately help fight against the effect of climate change and wildlife diseases.
Drivers to explore are temperature variables, precipitation, humidity, and extreme events. New models are also needed to combine multiple principles to accurately analyze and predict how, where, and when diseases may arise due to climate change. In some systems, we can readily see and detect impacts of disease, but struggle to do so in others. In oceans, we can see impacts of disease on corals, oysters, and abalones, and in terrestrial systems, we can see impacts on plant disease and some wildlife. But for many human diseases, and with highly mobile species, detecting effects of climate change remains a problem. Understanding the interactions between climate and disease is important to grasp, as the research done can be applied to multiple environments around the world, helping to predict future interactions and how we can battle them.
This is our works cited page for our outside resource used for the first introduction slide.